Welcome back. In the previous two lessons, we've learned that proteins are strings of amino acid building blocks that self-assemble into complex 3D structures, and that the human body makes hundreds of thousands of different proteins. Further, different cell types make different sets of proteins to enable each cell type to perform its specific function. Thus, your liver cells contain a different set of proteins than your muscle cells. But how does each cell type know which proteins to make and how much? Where do the instructions come from? You will learn the answers to these questions and more in this lesson on mRNA structure. Please take a moment to review the learning objectives for this lesson. Going back to our building analogy, the instructions for creating a building are in the form of blueprints. The same is true for proteins. The blueprints for proteins are called messenger RNAs, or mRNAs for short. As their name implies, messenger RNAs carry messages for how to make proteins. Now, let's watch a short video to learn more about mRNAs and their structure. Like proteins, mRNAs can fold up into complex structures. This is because the nitrogenous bases can pair with one another to form a double strand. These double-stranded regions intersperse with single-stranded regions to form complex floppy structures. We can directly visualize mRNA structures using a technique called atomic force microscopy, or AFM. The principle of AFM is really quite simple. It involves dragging a very, very fine needle across a surface on which is deposited the tiny object one wants to image. As the needle is dragged across the surface, it has to move first up and then down to get past the object. The different heights are then detected by a laser and recorded. With repeated passes, a three-dimensional image develops. This image shows three identical 883 nucleotide mRNA molecules that contain the blueprints for green fluorescent protein, or GFP, a protein that makes jellyfish glow under UV light. What you can see is that even though they have exactly the same pattern of A, C, G, and U, that is, they have the same nucleotide sequence, each individual molecule folds in a different way. This next image shows many more molecules of different mRNAs that contain the blueprints for GFP, for firefly luciferase, which is the protein that enables fireflies to glow, or the SARS-CoV-2 spike protein. In fact, this last mRNA is exactly the mRNA sequence that went into Moderna's original COVID-19 vaccine. So what you can see is that mRNAs are floppy string-like molecules that fold up into a variety of structures. In many ways, these structures resemble those formed by old-fashioned telephone cords. So now we understand what mRNAs are and their basic structure. In the next lesson, we'll learn how the information stored in mRNA is decoded to create proteins.